Recording. Share the screen. Might check my volume. Let's turn that up a little. Did anybody say anything? That volume was way down, so I might not have heard you. I don't think I need that. All right, so we'll finish chapter 3 today. And then that's it. Next week we'll start chapter 4. Next week there will be a quiz due on Wednesday, quiz 2. It'll cover labs 3 and 4, chapter 3, and the end of chapter 2. So the last half of chapter 2. There is no lab today. You can work on lab 4 if you haven't finished it. I will show up at 6:30, but I'll only stay for about 15 minutes unless there are questions. So at 6:45, if there's no questions, I'm going to log off. All right? We might have a short day today. We were talking about the Gram stain, and I discussed this slide showing you the different colors of all the cells in the Gram stain. Initially, when you stain with crystal violet, all the stains stain purple. When you, not stain, but you apply the iodine, it's a mordant. The stain is still purple, but it is a more intense purple. That's not shown here. It's also slightly darker. And then when you decolorize, the gram-negative cells become clear. The gram-positive cells retain the crystal violet and they're purple. And then we stain with safranin, the counter stain, and the gram-negative cells stain pink. The gram-positive cells retain the purple color, and they do get the safranin. However, to our eyes, they will look purple because purple is a more intense stain, and that's what our eye focuses in on. If you were to look at the cells with the electron microscope, you would see that it has both stains in it. So there'd be one, if this is the entire cell here, there would be one region which would be pink and one region which would be purple, and there'd be lots of regions in the cell. Any questions about any of that? All right, so let's talk about the Gram staining procedure in depth. So we're just going to go over this again, and you are expected to know the Gram stain and the procedure. First, this is not, the first two steps are not part of the Gram stain, but there's something you have to do before you can Gram stain. You do have to make a bacterial smear on a glass slide. And for the Gram stain, you especially want it to be a very thin smear. If you have cells on top of cells, the Gram stain will not work well. The bottom cells may have trouble getting the stains in them. And then they'll also have trouble decolorizing if they're gram-negative cells on the bottom. After you make the bacterial smear, you then air dry it and heat fix it. That is important because if you don't do that, the cells will wash off. Particularly during the water washing steps of the Gram stain. But even applying the dye, you will lose some cells just by applying the dye. So usually you always do a bacterial smear when you're going to stain cells. The exception might be a negative stain, but we won't go into the exception. And the reason is that even if you're just putting on a stain, that will remove some of the cells. And then the water washing will tend to remove all the cells. So you must do the air drying, the heat fixing. You then flood the slide with crystal violet. This is the primary stain. It will color all cells, and it's the first one used, why it's called the primary stain. You apply that for 60 seconds or 1 minute, whatever one you want to use. and the crystal violet sticks to the peptidoglycan in the bacterial cell wall. All cells, both the gram-positive cells and the gram-negative cells, stain purple after the application of the crystal violet. You then lift up the slide just to get the dye off of it and water wash it. You know, I'm not sure why that isn't in the previous step there. Oh, there it is. It was in there. I just didn't say it. After staining, you then do the water wash to remove the excess crystal violet. And then you apply iodine over the slide. Iodine is a mordant. It increases the affinity of the crystal violet with the specimen. What iodine does is it hooks or complexes with the crystal violet. So now we have a larger molecule giving the cells color. And the iodine is applied for 60 seconds or 1 minute. You then flip the slide up, getting rid of the excess iodine, and then water wash it. Okay, any questions about that? After applying the mordant iodine, the color of both gram-positive cells and gram-negative cells is still purple, but it is more intense. I think I talked about the water wash. If I didn't, there's a water wash after each step in the gram stain. You then apply the acetone ethanol decolorizing agent. This washes the crystal violet out of the gram-negative cells, but not out of the gram-positive cells, as long as you do not overwash. If you do overwash, you will wash the crystal violet iodine complex out of the gram-positive cells. So you want to make sure that you don't wash for more than about 5 seconds. The best way to do it is to hold up the slide, add the decolorizing agent, and when the decolorizing agent stops, the color stops running on the slide, that's when you stop adding the decolorizing agent. And it's about 5 seconds. It's considered the differential step in the gram stain because at this point the gram-negative cells will be clear and the gram-positive cells will be purple. The decolorizing step is the most difficult because if you don't do it long enough, you will not wash out all of the crystal violet and iodine from the gram-negative cells. But yet if you do it too long, you will wash out the crystal violet and the iodine from the gram-positive cells. Is there any question about that? Now, after each step in the gram stain, it isn't important that you water wash immediately, because if you were to leave the stain, like the crystal violet, on longer, it's not going to do you any harm, assuming that it doesn't dry out on the slide. Obviously, that would be a problem. But as long as it doesn't dry out, it's not a problem. But when you flood with the acetone ethanol, you need to immediately water wash, because even the trace amounts of the decolorizing agent on the slide, I'm trying to show you a slide here, which isn't a slide, I'm using it as a slide. Even the trace amounts on the slide will continue to decolorize the cells. So if you were to leave that on, the gram-positive cells, if you give it enough time, will be decolorized, meaning the decolorizing agent will pull out the crystal violet from the gram-positive cells. So you need to water wash immediately. Any questions about that? And then the color of the cells, the gram-positive cells will be purple, the gram-negative cells will be colorless. The next step is to apply safranin. Flood the cell with safranin and you apply that for 1 to 2 minutes. So one easy way to remember the gram stain is that if you put 1 minute for each application except for the decolorizing agent, then you're fine. Safranin is a pink dye. It does stain all the bacteria. It's a counter stain because it has a different color than the primary stain. So it's pink and crystal violet is purple. After the 1 minute to 2 minutes is up, you then lift the slide, remove the excess safranin, and then water wash the slide. The color of the gram-positive cells, they will continue to look purple. Even though there is safranin in the cells, they will look purple from the crystal violet. And it's just that our eyes focus in on the more intense color. If you look at the gram-negative cells, they will look pink. The slide is then blotted with bibulous paper, and it's too bad you won't be able to do this. Pretend this is the slide. This is the bibulous paper. You just blot it, fold it over, and you do not rub. Rubbing like this. I don't know if you can see that. You just blot it. You can press down on the paper on top of it as long as you don't move the paper on the slide. That does not remove at least most of the cells, and it removes most of the water. The droplets of water that are left on the slide, and I'm not talking about droplets, I'm talking about micro droplets. They will not interfere with your viewing, and they will air dry very quickly. And if you apply oil to the slide, those micro droplets won't cause any problem even with the oil. So you blot the slide with bibulous paper, and then you can examine it under the microscope, including with oil under the 100x oil immersion objective. Any questions about any of that? That is the gram stain procedure in depth. So the gram-positive cells are purple after the gram stain. The gram-negative cells are pink. Why? What's the basis for the differential staining? The difference is related to the differences in the cell wall structure. The gram-positive bacteria have a very thick peptidoglycan layer. We'll talk a little bit more about what peptidoglycan is in a future lesson. You'll see it a little bit in just a minute, the next slide. But it's a disaccharide and amino acids. The gram-negative cells, or at least the bacteria, do not have that thick peptidoglycan. Instead, they have, if they're bacteria, they have a thin peptidoglycan layer in their cell wall. The gram-negative cells also contain a lipopolysaccharide layer as part of their cell wall, and I'll show you that in this slide here. Let me blow this up so it's viewed better. Of course, now you can't read it there. So the gram-positive cells we're looking at here, the yellow is their the cell membrane, and they have a very thick peptidoglycan layer for their cell wall. If you look at that, how do you say that? With an electron microscope, I guess, you would see that the cell membrane is a phospholipid bilayer with other molecules in it. We're not going to talk about those. And that the cell wall is composed of peptidoglycan, which is strands of it running on the cell, and there's strands next to each other. I can't see this very well. I have my glasses here. Not a good thing for me not to have my glasses. There it is. So there what do you want to call that? Strands of it next to each other, and then there's strands on top of that, And there's many more than 3 strands. This is just trying to illustrate it. The peptidoglycan is layer upon layer, strand after strand, and one of the strands is peptidoglycan. It's hooked together with amino acids, which are part of the peptidoglycan, and we'll talk about that more, I think, in the next lesson. I don't think we're talking about it today. So remember, it's more than 3 layers. You're only seeing 3 layers in this picture that comes from your textbook, but it's many layers upon layers, and it's a very thick peptidoglycan layer. And there isn't a whole lot more to that cell wall. Peptidoglycan, and there are 2 other molecules, one of them shown here. We're not going to talk about it now. We'll talk about it later in the lecture, in another lesson. That's different from the gram-negative cells. When we look here, it has a cell wall and a cell membrane. The bottom yellow is the cell membrane. And the cell wall is right here with purple and yellow. The purple is a very thin layer of peptidoglycan. When we look at that under the electron microscope, you would notice that it is rows of peptidoglycan, one after the other. And it's only showing you 1 layer of peptidoglycan. That's not correct. It's a thin layer, but it is layer upon layer. I don't know how many layers, but it's many less layers in the gram-negative cell than in the gram-positive. So the important thing to remember is it's a thin peptidoglycan layer, and it is not just 1 layer. One, what do you want to call it? 1 plane with peptidoglycan strings running on it, meaning that these strings are layered upon each other, but they're much thinner than the gram-positive cell. And the gram-negative cells also have a lipid bilayer on the outside of the cell wall. It's called the outer membrane of the cell wall. And besides that, it's a lipopolysaccharide, meaning that it has lipids, and at least one of the lipids has a polysaccharide, meaning a chain of sugars in a polysaccharide hooked onto the lipid, so it's a lipopolysaccharide. The important thing to realize is that some of these lipids in the outer layer of the bilayer contains lipid A, and so it's these blue parts there. That's the lipid A. Why lipid A is important is lipid A is an endotoxin. Toxins are substances which are toxic to us or poisonous to us, and so any exposure to a toxin is obviously a bad thing. And gram-negative cells have an endotoxin in their cell wall. Any question about any of that? We'll talk about endotoxins in 2 other times in the lecture. Let's talk just a little bit more about this outer membrane of the cell wall in the gram-negative cells. You'll note that this entire lower level, meaning the lower level of the bilayer of the outer membrane, looks familiar. It looks like the phospholipids of the cell membrane. And that's what it mainly is. There are other molecules in it, but the lower layer of the outer membrane of the cell wall is mostly composed of phospholipids. The outer layer of the outer membrane of the cell wall does have phospholipids. Those are the yellow ones right there. But it also has these blue lipids, which are lipid A. All right, any questions about that? There are other molecules in the outer membrane, like the boron molecule here, and then you can see another one there. Is that labeled? Yeah, it's not labeled. All right, any questions about any of that? So it's this thick peptidoglycan layer that catches the crystal violet iodine complex, and it does not wash out in the decolorizing step. And the thin peptidoglycan layer, the crystal violet iodine complex, does wash out of it in the decolorizing step. And that's why there are gram-negative cells and gram-positive cells. Any questions about that? I've already talked about that. And I've talked about that. So let's look at a gram stain that has many different types of cells in there. You can see a gram-positive rod or bacilli right here. That's a coccus. Can anyone tell the color of that one? Let me blow this up. That looks like a gram-positive coccus. And there's a gram-negative spirilla. Anyone see a gram-negative rod? There's a gram-negative rod. And then there are some gram-positive, either cocci or bacilli. There's about the same thing. Those look like cocci, gram-positive cocci right there. All right, any questions? If not, let's move on. The gram stain is not so easy to interpret. Like if you were to get a slide like this, how do you read that? Well, there are bad regions in a slide and there's good regions in a slide. Use the good regions of the slide to make your reading on the gram stain. Let me blow that up a little bit. It might not be totally apparent, but you'll notice that there's a darker staining around here. What this is, and that, and there's also darker staining around this here, is that these are crystals of crystal violet. And I know that's a little confusing, but crystal violet in the solution can come out of solution and precipitate as a crystal. And then crystals can form in the bottle. And then when you go to use it, the crystal can come out. And the crystal won't stain the slide correctly. Like I said, sometimes I've seen on gram-negative cells, and there will be a crystal there. Around the region where the crystal is, it's like the crystal violet is releasing, the crystal of the crystal violet is releasing the stain, and so it will then color these cells purple, even though the rest of the slide is pink. And I'm not sure that that's really pink there. Anyone think that's pink? It's lighter than here, but I'm not sure that that's pink. And that's just the way this photograph is. It's not clear to me. Is that pink in color or is that purple? I'm not positive. If anyone can tell, that's fine. But my point is, you don't read and interpret your results around this region and this region and this region. You look at where it's a good region of the slide, easy to distinguish individual cells and to determine the color. Any questions about that? The gram stain is not, is not always clear. Sometimes the cells will give you a problem, sometimes the stain will give you a problem, and so you have to do some things to make sure that the gram stain is valid. The first and most important thing is always use freshly grown cells when you're going to do the gram stain. Because if you have old cells, they may not stain correctly, particularly if they're gram-negative cells. That is, the dead cells do not stain correctly in the gram stain. And so you want freshly grown cells. That means cells that have grown for 2 days or less. And the gram stain is not universally applicable. Some bacterial cells stain poorly, if at all. That's particularly true for the Mycobacterium. They just do not readily take up the stains of the gram stain, meaning the crystal violet and the safranin. And the reason is that crystal violet and safranin are water-soluble stains, and the Mycobacteria family have so many lipids of macolic acid in their cell wall, they do not readily take up the stains. So the results are not always as clear-cut as the theory would suggest. And this will be more apparent to you when you're working on your unknown project. You'll be given a slide, and sometimes it could cause you problems. I'm not trying to be negative here, but when you're given the gram stain on your unknown, there might be regions of it that will be not staining as well as the good regions of the slide. And I won't ever give you a slide that'll show you the wrong color. I'm not going to do that and expect you to realize, maybe it's not quite correct. But another thing is, when you're doing the gram stain, the growth arrangement may not be applicable. Like right here. Oops, trying to show this a little better. This looks like it is a streptococci. But then right in this region, or that region, you could argue that it's a staphylococci. And is that a diplococci? Let me blow that up a little. Yep, that's a diplococci right there. When you're doing the gram stain, if you want to determine the cellular growth arrangement, you have to grow the cells in a liquid culture, because otherwise they may stick together. This one is not really sticking together that badly. Maybe this was grown in a liquid culture, because usually when you grow them on a petri dish, the cells stick together and you cannot tell the cellular growth arrangement. Any question about that? Oops, didn't mean to do that. All right, let's move on. The structure of the gram-positive and gram cell walls not only affects the staining of these cells, it also relates to how they respond to certain antibiotics. For example, the gram-positive bacteria tend to be easily killed with the penicillin G antibiotic, as well as cephalosporin and many detergents. So the gram-positive bacteria are easier to kill by certain antibiotics like penicillin G and detergents, whereas the gram-negative bacteria are resistant to these antimicrobial agents. The reason is the gram-negative bacteria have an outer membrane in their cell wall which prevents those antimicrobial agents from getting into the cell wall. This outer membrane of the cell wall can restrict molecules from getting through it, and penicillin G cannot get through that outer membrane of a gram-negative cell. Any question about that? And that's why penicillin G works very well on gram-positive cells, But most gram-negative cells, the penicillin G cannot get through, cannot get through the outer membrane. And it will not harm the cell, the bacteria. What you would do if somebody had a gram-negative infection is you don't use the antibiotic penicillin G. You would use an antibiotic that could get through this outer membrane of the cell wall. And there are a number of them. Actually, even in the penicillin family, like ampicillin, which was a really early member of the family to be developed. I think that antibiotic came out in the 1960s. So ampicillin and carboxycillin are just two of the antibiotics. And then you could use an antibiotic which is lipid soluble. And I'm trying to remember a lipid soluble antibiotics, and I'm drawing a blank here. I think it starts with an S, but I can't remember the name. But there are many antibiotics that can be used on gram-negative cells, like tetracycline, works on both gram-positive and gram-negatives. Does anybody know an antibiotic that you use for mycobacterium infection, like Mycobacterium pneumoniae? Does anybody know what that antibiotic is? I'm drawing a blank here on what that antibiotic is.
I mean, there's several.
I know, usually 4 are given, but one of them in particular is a common antibiotic, and that's the one I was trying to remember.
I think ampicillin's one of them.
Ampicillin could be used. Do you know another?
There's ampicillin, there's cephlexin.
Clindamycin?
Clindamycin. No, that's still not the one I'm going to. Anyways, I'll just have to move on. I don't remember. All of those do work on gram-negative cells, but I was thinking it was beginning with an S, but I'll have to look that up. Anyways, not important. The point is that you can treat gram-negative cells. You just have to use it with an antibiotic that can get through the outer membrane, and there are a large number. At least half of the members of the penicillin family can be used to treat gram-negative cells. And I don't know, there's 40 or 50 members of the penicillin family. Okay, but penicillin G does not get through the outer membrane of the gram-negative cell wall. And the only other antibiotic I know that does not is penicillin V. That was the second antibiotic ever discovered. Not second antibiotic of the penicillin family, I should say. All right. We've already talked about that, so I don't need to talk about it further. Sorry, I was here and I moved back to show you that slide. So, are we done here? Yep. So, 2 last slides, I think, that I want to talk about. You won't be quizzed on these slides, but I want to talk about them because you may need the capsule stain when you are working on your unknown project in the lab. And so I just want to talk a little bit about the capsule stain. What it does is it stains the background and the cells, but not the capsule. And you may need to use this to help identify your unknown bacteria, which you will be given later in the class. Let me put that right here. Yeah, that's good enough. So when you stain, you can see a clear region around the cells. That's the cell capsule. There's the cell. And then this stain is staining the background of the cells as well as the cells. And this is probably a differential stain that's used for showing the capsule. Another stain which is even easier, which is a special stain, a negative stain, India ink, and it stains the background very dark. And as you can see, it does lightly stain the cells, so you can see the cells, and then the capsule is unstained. Okay, any questions about the negative stain or the capsule stain? All right, if there's no questions, we're done. This is really early. So, work on your in-class assignment for this lecture. There's, I think, 2 worksheets in the lecture that are due this Saturday. Work on those. And I will see you at 6:30 if you're coming into the lab. And remember, I will only be there from 6:30 to 6:45 unless there are questions.
Cool. Thank you.
All right. Bye.